Hello, class. So welcome to genetics. So this is going to be our intro to genetics lecture one. Um, I'm Daniel McCardle. I'm going to be your professor for this. This is absolutely not how I intended this class to go, but we're going to roll with the punches that we have. So here we are, getting ready. So today we're going to talk about essentially what is genetics, right? This is going to be our big picture topic that we're going to kind of look at. And essentially, it's very much genetics is just biological inheritance and all the various forms and ways in which these are going to affect both our understanding of ourselves and kind of life in general, right? There's going to be a few ways that we are going to look at genetics, right? The first one that we're going to kind of get tucked in on is this thing called rise of genetics. There's a couple of different modes and methods that we're going to do. Um, and each of these are going to be kind of foundational to our overall understanding. What we have right now is the rise of genetics in pangenesis. So pangenesis, right? These are the two things that we're going to do, right? And they are very much based and passed from embryo at conception. And what they do is they carry information from parts of the body to then the reproductive organs. And this is very much led to the theory understanding of inheritance of acquired characteristics, right? We will kind of touch on what these mean in a later sense, but for now it is kind of very important that we at least get some of the theories down. Next up, we have preformatism, right? And these are very much that we have, that we as individuals are very much miniaturized adults once we have finished development. And then from there, these traits are going to be inherited from parents. Next up are their next two, right? We have blending inheritance and then germplasm theories. Blending inheritance is very much that each of the individual offsprings are going to be a blend of both parents' genetic material. And it gets harder to separate them out into the later and future generations. And the last one is germplasm theory, where cells carry a complete set of overall genetic information. Right? So those are the four. We have a couple of things that will be interesting for us, which is very much our areas overall of genetic study. What we will do is how they will be classified for us will kind of determine what they do in the future portion of our class. So first up, we have classical genetics, which is very much concerned with overall gene location and the behavior of those chromosomes on that gene location, right? This was originally proposed by Mendel. He's going to be this guy that we talk about a lot of, Gregor Mendel, and we will get into why he was important, what he did, why we still talk about him. But his understanding was that each organism is going to have two units of traits. These are usually going to be determined as X and Y, and we will kind of talk about that, why he kind of decided on X and Y, why they remain interesting. But this is very much this idea of classical genetics, right? There's then two laws of inheritance, and then ideally individual genes are very much discrete units on the chromosome, meaning you have a gene for eye color, but it is just one gene. There's not seven different genes for eye colors that interact. It is very much just a one gene type of situation. And then overall, those genes are going to produce things that we will call enzymes. Next up is this idea of molecular genetics. Those are the very much determining for structure and control of the material. So it's less concerned about the individual gene and more control of how, more interested in how those genes are controlled. So DNA is going to be the thing that we are going to talk about an awful lot of, right? That is very much our genetic material. Mutations are going to alter that material. And then this next one is replication, transcription, and translation. And that is very much how we get the material from one person to another, right? Each of them are going to be, touched on in much greater detail in later in the course. But for now, it is very helpful to understand that that's kind of where it starts. And there's individually, we are going to talk about an awful lot of like diseases, what they do, how those genes are causing those diseases, and all the various associated things. And then next up, we have genome sequences. So each of them are going to be fairly helpful. And then we have evolutionary genetics. Evolutionary genetics are very much the mechanisms of evolution in gene changes, right? So this is very much how those genes, not interested in what they do or how they control it, but it's very much the viewpoint that genes will change over time, and how do we then measure that, right? Two people did a ton of work on this particular thing of evolutionary genetics. Those were Hardy and Weinberg, right? We will talk an awful lot about them because they have done some very foundational work, and we will reference their work pretty heavily in this course, right? So Hardy and Weinberg, right? Both of those are two things. And what they officially did was they developed some rules for us to say that no change is happening. So if Hardy and Weinberg are right, and so the idea is if that mechanism essentially and that equation equals 0, then there is no change. Frequently, there is very much a change that happens, and we need to kind of look at that. And then lastly, there's then social behavior. So this is very much how the individuals of populations are going to kind of interact, and this is very much how those genes work in overall goals, and we'll talk an awful lot about them later in the course. So just as a way of kind of backing everybody up, right? This is the possibility that we've not taken a science class in quite some time, right? We will use the scientific method. And that is very much like a rinse and repeat type of process, but it is very interestingly, it's a way to know. So it's a way of us to actually acquire overall knowledge and have a very consistent viewpoint on that. We are going to use essentially deducting. So deduction is essentially, it's defined as finding data to support an argument. Um, when you actually have to use data, right? It's kind of this nice little overall goal, and it's almost like a waterfall type deal where you start at the top or the bottom, really doesn't matter. And what you do is you build out that, but you always have this way to go back and understand a little bit more. So the steps are observation and then hypothesis, and then from there you are going to predict what we are going to try to see. Then we're going to test that overall hypothesis, come to a conclusion, and then lastly, this idea of understanding, does it really fit? So universality is very much what we're going to do is we are going to test our discovered phenomenon, aka our conclusion, and see if it actually still continues to fit. We are going to jump into this last section of essentially how do we actually do genetic research. So this class is going to be very different from a lot of the other classes that I've taught, which is almost exclusively online with the exception of a few in-person labs. And so these labs are going to be foundational to us understanding how do we actually do genetic research, right? We are going to need to choose an organism that's going to be pretty idyllic, right? So it's ideal traits for genetic research. Some of them that we might particularly use in later in your educational field might be, Drosophila melanogaster, essentially fruit flies, right? They're pretty easy to culture. They grow very easily. There's very large chromosomes, and they generally live for about 2 weeks, right? So they have a fairly short lifespan, meaning you can breed an awful lot of them, and you can kind of go pretty steadily on. The next one, especially for those of you that are going on and will be taking microbiology, the big main organism they use there is Escherichia coli, so E. coli, right? The reason a lot of microbiologists use it is very much because it has a lifespan of about 20 minutes. It has a very small gene number, so a very small number of genes that actually control the actual individual bacteria itself, and it grows very easily. So they love it to kind of go in. And again, because it has a very short lifespan, so you can do a ton of research on E. coli. The last two are one that we will use, right? We will use number 4, which is yeast. It is a very simple structure. It's very easily manipulated and can be ready in a very short period of time, which is why we will use it. That is how we will do our genetic research in this class. In the news an awful lot now are viruses. They are a ton of subclass in there. We have a very, very simple structure. They can have, as we have found out, a ton of overall arching implications. But what they do is they readily repeat, which is kind of the reason that we have to do all of this class kind of online and not necessarily that is because they have a very, very, very quickly increased speed, meaning the virus itself will replicate 100-fold. So 1 times itself times another 100. So 1 times 1 times 100 in an hour, which means the virus itself can, reproduce much faster than anything in the body can kind of prevent it from doing that, which is why we have kind of the situation that we currently have. Viruses themselves are very, very scary and also um, very interesting to do genetic research on. And then the last thing is essentially, is this class really widely usable, right? Everybody wants to understand what they're doing and why, but we really need to know, is it actually useful? Is it worth your time? I would postulate yes, because we do a ton of stuff in genetics. And this is just kind of some, very, very narrow listing where, some of it has to do with things that we do. So pharmacology, disease diagnosis, gene therapy, genetically modified organisms and conservation biology. All of these are just some things that genetics kind of feeds into and does. And for those that are deciding to go off to either medical school or veterinary medicine or pharmacology or anything else, industry, academia, all of these are going to be useful for it as a whole, right? And then lastly, we're going to kind of finish out the day with essentially this last section of which is applied versus basic science. So applied science versus basic science. And we are going to look at and kind of justify the use of science by its application, right? It's applied research. That's kind of how applied research is going to work. It's going to be very much the research itself, how it is used is going to be the reasoning and the justification for it, right? But and that's very helpful. Don't get me wrong. Like forensic psychology and forensic medicine would be very helpful, and is very helpful with its individual application. But the basis of that science is very much dependent upon the basic research that is actually then used to support that applied research. So our viewpoint for this class is very much going to be, we are going to focus on the basics, right? This is very much a foundational class. We need to have that understanding to make sure that we have a good overall goal so that when we actually get into our and a good overall understanding so that when we move into some of our more advanced and higher level classes, we have a very good basic understanding of things like genetics, right? That's how we're going to kind of look at all of this. I hope this was wildly helpful. If not, I'm very sorry. So the reason that I've recorded this is that you feel free to read it or watch it all again. But that's kind of where we're going to get to all of this class. All right. Thanks, y'all. Have a good day.